linking healthy soils and healthy people through the microbiome. I'll be focusing on how soil microbial communities shift with ecosystem condition, how they also contribute to human health. In particular, how microbiomes provide a tangible link between the health of environments and the health of soils to the health of humans. So what is a microbiome? It's a community of microorganisms living together in a particular habitat. Over recent decades, we've come to learn that humans are not really a standalone organism. We're a system that includes microbes, and those microbes are a major part of us. Almost all of these microbes ultimately come from the environment. Especially strong influences in shaping our microbiome, you might have heard, are how we're born, whether or not we're breastfed, our diet, and the actual surrounding environment. Because studies in animals and humans show that transplanting the gut microbiome can transfer disease, and it can also offer protection from disease. So medical researchers are looking into gut microbiome transplants to treat inflammatory bowel diseases, irritable bowel syndrome, obesity, diabetes, cancer, fatty liver disease, multi-drug resistance, depression, neurological disorders, among other diseases. So what are some things to look for in a healthy microbiome in our gut and perhaps also in soils? Here's some likely candidates to look for. There could be key taxa or organisms with known beneficial roles. So in soils, think of nitrogen-fixing bacteria or mycorrhizal fungi, things that you can go looking for and you notice when they're missing. Particular constituents or molecular compounds that are released when microbes die and they break up, they can be taken up and used in health-promoting pathways. If you hadn't heard, soils are estimated to be home to between 60 to, some estimates say, up to 99.9% of species on Earth, making soils the richest of environmental microbial reservoirs. With increasing above-ground diversity, there are more complex interactions below ground and greater diversity of habitats with all those root zones interacting with each other because they can't do everything they need on their own. Excessive fertiliser regimes can have a detrimental effect on soil microbiomes. With excessive nutrient, plants lose the incentive to invest in the roots, fungal or mycorrhizal associations. And we've heard this sort of notion quite a bit in this sort of community. So there may be options for farmers to save money by dropping back excessive fertiliser inputs and letting the microbiome do its thing. It's worth noting that physical and chemical conditions can either support or put a handbrake on the microbial communities. For example, high organic matter and clay content, such as the soil on the left there, is going to provide premium habitat, whereas acid or alkaline soils, salinity, can represent barriers to building diverse soil microbiomes. We've studied changes in soil microbial community composition in degraded ecosystems as they're restored towards natural reference states. The example here is from post-mining forest restoration right here in Western Australia. The y-axis is a measure of how similar the community is to a natural reference, and on the x-axis is the age in years since restoration planting. In these forest ecosystems, without sort of adding your biostimulants and that sort of thing, just letting natural regeneration take its course, it's taking several decades to return to natural levels of variability. We were looking to compare crops and pastures under biological focus versus standard district practice versus native vegetation from some South Australian farms. So the two-dimensional plots here are a way to visualise community scale differences. The closer they are, the more similar are those communities. Points that are away from each other have very different community composition. The colours there represent different groups, so orange is standard cropping, pink standard pasture, blue biological cropping, purple biological pasture, and then the green is the native vegetation. I want to spend some time further linking soil health and human health. That's the interconnected health of soils, ecosystems, plants, animals and humans. Over the last 20 to 30 years or so, a growing number of medical researchers and immunologists have been advancing the biodiversity hypothesis. This is the idea that loss of exposure to natural microbial diversity through biodiversity loss, through soil and land degradation and urbanisation are contributing to a rapid global rise in allergies, autoimmune and chronic inflammatory diseases, especially in urban areas where many have largely lost contact with soils. And soils from urban green spaces have become more homogenised globally due to similar green space types and management. These have become home to more fast-growing, opportunistic potential human pathogens and more antimicrobial resistance genes and communities better adapted to faster nutrient cycling and stress. In hospitals, generally speaking, we can probably agree that the only good microbe is a dead microbe. But this linear way of thinking is perhaps not the best template we should take into everyday life. Not enough exposure to naturally diverse range of microbes is leading to poor immune training and immune dysfunction, so we get negative health outcomes. This aligns really well with the generalizable U-shaped optimization curve that we see right across biology, this idea of shifting from deficiency to sufficiency to toxicity. The image here is a bit of an extreme example. The person has just prepared an animal. They're cleaning their hands with the microbe-rich stomach contents of the same animal. This person is probably building immune fitness, and they're using the microbial diversity to counteract potential pathogens. So these microbes that come from the environment, from soils, can find their way into the gut and help to boost our health through some key pathways. 2 studies here. On the left, studies of asthma and allergic risk in kids growing up on farms versus cities. You can guess what the outcome is here. It showed lower rates of asthma and allergy for the farm-exposed kids. Intervention studies with daycare children, where researchers brought soil and plant materials into daycare yards, found changes to their microbiomes and improved immune biomarkers for immune tolerance within 30 days. On the right panel, comparing traditional Amish farming communities with greater exposure to soils and animal manures versus Hutterites who employed more industrialised farming practices, showed lower rates of asthma and allergies in the Amish farm kids. And I found that in the lower socioeconomic groups where you expect to see environmental influences on health stand out the most, that hospitalisation rates for infectious and parasitic diseases, which are rising on the y-axis there, they were highest in regional local government areas with typically low microbial diversity soils. So these results are suggesting that areas with high microbial diversity soils may help boost the immune fitness of populations living there. Anxiety-reducing responses have also been found from exposures to the soil bacterium Mycobacterium vaccae. This study has previously gained a lot of exposure due to promoting mental health benefits for gardeners. The data here show potential metabolism of compounds on the y-axis across 3 different data sets. The data set with the lines is a soil metagenome data set from post-mining restoration, and the other 2 are gut metagenome data sets from type 2 diabetes versus healthy controls from Sweden and China. In the most degraded soils, which correspond to the red and blue arrows, and in type 2 diabetes gut, they shared elevated potential metabolism for certain sugars and decreased potential metabolism for lignin or plant-based fibre. Also decreased potential metabolism for an interesting group of molecules called branched-chain fatty acids. What might be going on? If we compare intact ecosystems on the left versus degraded ecosystems on the right, generally speaking, there'll be larger reservoirs of soil organic matter in intact systems, more plant debris, including lignin-type fibres, and more recycling mechanisms. In degraded systems, there's typically a loss of soil organic matter, which ultimately breaks down into simpler and simpler sugars. This could explain the increased potential metabolism of sugars and the decreased potential metabolism of lignin, which might be transferring an altered metabolic profile to the human gut as people experience greater exposure to degraded ecosystems. Branched-chain fatty acids are really precious molecules. They're recycled and transferred across bacteria, fungi, and animal domains. The stuff that's released from soil organic matter becomes a key ingredient to make these branched-chain fatty acids, and they're also recycled and lengthened as they're recovered, for example, through fungal recycling networks. In the gut, these fatty acids are taken up by human host cells, where they're involved in lipid metabolism and the central regulation of human host cells. They're actually needed to trigger a particular mechanism called the mechanistic target of rapamycin complex 1. That's a mouthful and it sounds super complicated, but the important thing is that when that thing is not working, it's related to diseases, including diabetes, cancer, and neurodegenerative disorders. So regenerative practices that focus on building and protecting soil organic matter and soil biodiversity will shift microbial communities and their functional capacities. Biodiversity and reduced disturbance are associated with greater health protection and progress towards One Health approaches. Soil and gut microbiomes have a degree of overlap. That actual degree of overlap is yet to be fully defined. Exposure to naturally diverse soil plant microbiomes show health benefits through immune training, seeding, shaping, and resupplying gut microbiome and functions. Hopefully raised awareness of the scope of soil and gut microbiome connections and the linking of healthy soils to healthy people. Thank you.